Let's look at some chemicals that might interfere with nerve transmission. In the past, we talked about agents that act at the neuromuscular junction. For example, Botox blocks the release of acetylcholine from the end bulbs. There's also other things, either in nature or chemically synthesized as drugs, that block not at the end bulb, but at the actual action potential step. And some of these we've heard of. We've probably experienced local anesthetics before. Local anesthetics are used to anesthetize the skin if you're getting maybe a stitch for a cut, or a lot of dental procedures are numbed using local anesthesia. And some examples you might have heard of are Novocaine and lidocaine. Well, those drugs, those chemicals, actually block the voltage-gated sodium channels at the sensory neurons. So if you put enough of these drugs in areas of the skin that are getting sutured, all those pain neurons can't transmit their action potentials to the brain. And if they can't transmit those action potentials, you do not perceive that pain. So that's a very useful clinical connection to some of the things we've talked about. Some other things can be found in nature that interfere with neuron function. There's a lot of organisms, as you can imagine, they protect themselves by having toxic substances. One example is a frog called the poison dart frog, found in Central and South America. It produces a toxin that actually will do a similar thing and block voltage-gated sodium channels. Problem with this is if it goes systemically, you can interfere with a lot of neurons that way. There's pufferfish and triggerfish that are found in parts of the world that produce a neurotoxin. Other neurotoxins can be found that block other types of channels. For example, scorpions produce a potassium channel blocker. So they interfere with not the sodium, but the potassium channels. The black mamba snake, which I have a picture of over here, stay away from him. His neurotoxin is a calcium channel blocker. So a lot of organisms out there have figured out ways to protect themselves by blocking neurotransmission in either their prey or some predator that might want to eat them. So try to stay away from these guys if you come across them. This figure we've seen in the past when we studied the neuromuscular junction. The neuromuscular junction is an example of a chemical synapse. A synapse being a connection between two cells. The neuromuscular junction, the connection was between a neuron and a muscle cell. We also have connections between neurons. So this would be one neuron that must have been stimulated somehow, and it is communicating and possibly stimulating this second neuron called the postsynaptic neuron. So what we have is the synapse here, and if you're before the synapse, if you're the one releasing the neurotransmitter, you're the presynaptic neuron. If you're the one responding to the neurotransmitter, you're the postsynaptic neuron. And in between those two neurons would be the neurotransmitter communicating from the pre to the postsynaptic neuron. There are other synapses. There's actually a type of synapse called an electrical synapse, where there's no need for the neurotransmitter, and the action potential can actually flow directly from one cell into the other. But we're going to stick with the chemical synapses. And again, if you understood the neuromuscular junction, you'll understand these chemical synapses. A lot of the same players. We have the nerve impulse coming down, opening the calcium channels, voltage-gated calcium channels. That calcium is going to be involved and required for exocytosis. We get exocytosis of the neurotransmitter, which binds to a ligand-gated receptor channel, and then that's going to allow depolarization of the postsynaptic neuron. And then that neuron, if it reaches threshold, will produce its own nerve impulse. There are also examples where maybe this presynaptic neuron is inhibiting the postsynaptic neuron. So sometimes we need that type of response. In order to inhibit the postsynaptic neuron, you don't want to reach threshold, you want to get farther from threshold. And we call that hyperpolarization. Then it gets even more complicated in the nervous system in that quite often you don't have just a single presynaptic neuron. You might have many presynaptic neurons that all are releasing neurotransmitters that have to be assessed by the postsynaptic neuron. We call this kind of assessment of all these graded potentials net summation. So net summation is when this neuron has to kind of take all these little graded potentials that are occurring at its dendrites into consideration and determine whether it's going to fire or not. And again, that's largely based on reaching threshold or not reaching threshold. So notice that some of these neurons, like presynaptic neuron 2 and 4, are releasing what's called inhibitory neurotransmitters. Those inhibitory neurotransmitters aren't going to be depolarizing, they're going to be hyperpolarizing, compared to the excitatory neurotransmitters shown in red, which would be more of a depolarizing effect. So that's one way we can classify graded potentials, but also the neurotransmitters that are causing the graded potentials. If the neurotransmitter is causing a depolarization, it's an excitatory. So these are called excitatory postsynaptic potentials. Usually that's caused by sodium influx. If sodium goes in, you become less negative or more positive, which we call depolarization. If the neurotransmitter hyperpolarizes, that produces an inhibitory postsynaptic potential, which is usually bringing chloride in, which is negative. So you bring chloride in, you become more negative. So now instead of being maybe negative 70, you drop down to negative 80. That's called hyperpolarization. Another way to become negative is to get rid of more positive. So when potassium goes out, that's another way to hyperpolarize the membrane. So when we get to neurotransmitters, we're going to be using, terms like excitatory or inhibitory based on what do those neurotransmitters do to the cell that is responding to it. There's a couple types of net summation. One is known as spatial summation. That is when many graded potentials occur at the same time, but maybe at different locations, different dendrites. And that neuron sums that, right? They kind of add. So on this graph, we've got, here number 1 represents a graded potential somewhere. Here's another graded potential. But then if you have many graded potentials occurring at different dendrites, that all adds up. They're additive. They sum. And this is telling us that because there were many graded potentials occurring even at different locations, it reached threshold and caused the action potential. That's called spatial summation. Temporal summation is when you have many graded potentials but at the same location. That also adds up. So here's one, causes a graded potential. There's another one. But here you got fire, 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 and they're adding up and reaching threshold. So the difference is, are the graded potentials at different locations or is it at the same location? Next video, we're going to run through the different neurotransmitters in the body. There's about 50 of them. We're not going to learn 50 neurotransmitters, but we're going to learn, some of the more common and some of the more important neurotransmitters for when you get into medicine.